Hello everyone, this is Mrs. Travai and this is Chapter 7, Blast N, and these PowerPoints have been provided by the Rutgers Waxman Student Scholars Program. Okay, let's get started. So we've gone through here and now we're at this point where we're going to search the database to see if there are any matches to our sequence. And now we get to this Blast N page on DSAP. And again, if you get confused, there's this life preserver that you can click on and you can get some videos or information for help. But what you're going to need to do is retrieve your sequence. So click on here and copy the sequence. And then you're going to click on here and this will take you to NCBI. And you're going to be looking at two different Blast N databases, the NRNT database and then the Blast N EST database. Make sure to scroll all the way down this page because many people miss this part and don't understand why the sequences have errors or the form has an error at the end. So once you get to NCBI and you get to their BLAST page, this is their homepage for BLAST, and the link actually brings you directly to this nucleotide BLAST. This is a basic local alignment search tool, and this was developed using algorithms to be able to analyze and match the best possible matches to sequences that you run in this program. So here you can see that there are tons of sequences on the database, and again, this is where you can publish your sequence when we're done. So let's get going. We're going to take a look at the Blast N search page. You're going to enter your information into this box, the sequence, and I wouldn't touch these for now. When you get more experience in this program in the next year or so, maybe you play around with these, but right now we're just going to select Blast and retrieve our sequence, put it into the database, and select from the database, here where it says database, you're going to select the NRNT to start with. Your search options, again, are parameters that we're going to use the default. As the Blast N progress report goes through, it may take a few minutes to search through the entire database. And once you get your results, you can see a picture, a view, a representation of what your sequence is matching. So if you take a look here, here's your query. So this is your sequence 1 through 700. And it's showing me here that it's matching pretty well, if it's over 200 bases, in this region here, but we get no matches over here. You can also have a graphic report for example sequence 2, and you see that if you get little breaks, little sections, that the colors are going to be different. So it depends on how many bases in that region you're actually matching. The best matches are going to be listed at the top of this page when you scroll down on the search page, on the results page. And we take a look, and there's a lot of information here. We're going to take a look at each part and be able to understand what we're looking at. But right now, try to answer which match is the most meaningful or most significant for us. Number 2, which part of the gene appears to be the most conserved with other organisms? Okay, click for question 3. Are those matches at the end significant? Number 4, which of the following e-values indicates a best match? Now the e-value I didn't speak about before, but the e-value is the probability of actually matching something on accident. That's my simple way of explaining it. And if we match something on accident, we're going to get a high number, something like maybe 5.3. So we want it not to be on accident. So if you want your matches to be significant, which one would be the best? Now, there's a couple things that you get when you scroll down the page beyond the information and the actual line-by-line information of matches. You can see what are called the alignments. And if you scroll down on the page, you can see the length of the sequence, the match, the expect value, the e-value. Again, you want to be as close to 0 as possible. And this is pretty close. And it tells you here if it was identical. There was a 79% identity match, And were there any gaps in the sequence where if you put a dash in, it would actually fit better? And in this case, there's no gaps. But if there was a gap, you'd see dashes in the sequence. Our sequence is the query, and the sequence in which it matches in the database is called the subject. So here you have an area where there's a mismatch, right? Everywhere there's a little gap. This was a G, now this one has an A. Here you have a C, and that's a G. And areas that have lines between them indicate a match. Perfect but short matches are not very meaningful. And I talk about this when we do this in AP Biology, where we actually search a 15 amino acid protein. And if you remember, I said that we're going to get very poor expect values. Our expect values are going to be whole numbers, and numbers beyond, higher than 0, and that's not very good for our purposes. So in this case, although it matches perfectly, the expect value is high because it's so short. So examine the best alignments, and are they significant? And that's what we're trying to see. So could the mismatches be bad sequences on our part? Was it the machine read it and maybe it was incorrect? Bad sequencing on the part of the people who submitted their samples? Or is it that it's differences in two different organisms, and one organism has one variation and the other has the other? Remember that the DNA code is redundant, which means that there are multiple codons or multiple groups of bases that will code for the same amino acid. So even though you have a difference, let's say here, it's still coding for the same amino acid. So there's not a difference in the actual protein. So in this case, you see wobble positions, and you see the last base is different. And in this case, everything's fine, but the last base, and again, the last base. The third base seems to be the degenerate code, the area where more than one codon can read for the same amino acid, and it's due to what we call wobble. Small gaps can alter the reading frame. So when you put your sequence into NCBI, it'll actually incorporate in the algorithms, there's a way to see if there are possible gaps. And if the gaps, when placed into your sequence, into the query, actually make a better match. So it will give you that information as well. So an example of a match with and without gaps. So without the gaps, if you take a look here, so you have bases. Here's our cDNA. We have an exon, and here's an intron, exon. And there's an area here where you have some gaps, and it's actually reflected in the sequence. So you look at the alignments, and you want to look for an actual protein name. If you get hypotheticals, scroll down and see if there are actual protein names listed later, because you might get good matches, but they're just not the first ones. So here you see that the poly A tail is matching in this region. Does that mean that this is a match to the rest of our protein? No. If you could see, it doesn't match this whole area, but there is a gap here. So near the end, you're going to get matches just because of the poly A tail. Is this match biologically significant? Okay, take a look at it and see, does it look like it should be significant? You make the call. And again, we'll discuss this in class. 6, is this match biologically significant? And look at the expect values, look at your results. Okay, is this a protein coding region? What is the likely explanation for that gap? And again, these are difficult questions. I don't expect you, if you're having difficulty picking an answer, like I said in class, we're going to discuss this. Is this match likely in a protein coding region? Okay, so once you pass the region where you actually get some results, you're going to enter the information into the form. And I think this is probably the most, one of the more difficult parts of the program to get through. It's so easy to do, but it takes time. So in this case, we're going to put 3 different organisms, our best matches, but look down the list so that you can get the information that you need from 3 different organisms, not the same. And actually the DSAP form will give you an error if you put 3 of the same organisms. So on the clone report form, you can obtain some of the information. So when you click on the selection, it's going to bring you to a page on the clone report form for that clone. So if you take a look, the definition is there, the accession number that you selected from the list before. Then you'll have information on the organism and which organism it is. And sometimes it gives you the general name, the actual common name. But the E-value and all the other data, the alignments, the query start and query end are in this alignment here. So you click on here and it'll bring you down to this page, to this section. So when you're looking at this picture. And then if you select here, if you click on this entry, it'll take you to the clone report form that looks like this. So again, the information that you were looking for, the E-values and the query start and query end, are all in the alignment section. Okay, so take a moment to answer question number 10. And the third step is to perform the blast of the EST database. Now, express sequence tags were areas of the proteins that are non-coding that are of importance, and scientists want to look at them, but don't code for a protein. So typically we weren't putting them we were putting it all together in the database. They separated them into a separate section of the database. So in the blast end report, you have, for example, 1 sequence of the EST database. It matches the beginning. Here's your alignment, a pretty good expect value. There are no gaps, which is pretty good. And you fill out the table just as you did the other blast. So let's take a look at this last question, this question here. Why are there differences in the sequence? Okay, why are there some mismatches? Okay, and we look at the sequence and we take a look at where those mismatches could be occurring. And you should always look at your sequence to see, is that T here, okay, could it have been a C? Well, no, it's pretty obvious that it's a T. Okay, so then it must be that there's differences in organisms, maybe mutations. So it's similar to other organisms, but it's not exact. Is your cDNA similar to genes in other kingdoms? Now, if you scroll down the list, you might find that bacteria or protists or fungi in your matches, but and sometimes when we're working with plants, we only hit plants. So it's possible that you might find other organisms in different kingdoms, or you may not. That just means that those were your best matches. So is the sequence found in many other organisms? I mean, if you take a look down here, you find a lot of these entries, and all of a sudden it's all different organisms. Okay, so we know it's in humans, CMAs. So all these different organisms have a very similar sequence. And again, there's a big difference between the E values, but it's still significant. Okay, and hopefully this helps you analyze the DNA sequences. In class, we will be looking at your practice clones and discussing them while you're working on the computers. Okay, so have a nice day, everyone, and I will see you in class.